Welcome and thank you for joining. We give everyone a couple seconds to get connected to the audio, and maybe another couple seconds for folks that are joining a smidge late. But we'll get started in a minute. And for this session, Anthony is going to be doing the training. Thank you, Anthony. And if you have any questions, you can put them into the chat and I can try to answer them. And then also, if there's anything kind of pressing, we can pause and interrupt and ask the question in the moment. But feel free to drop stuff in the chat during the meeting. And also, please keep yourself muted unless you're unmuting to ask a question so that there's not too much background audio. And this session is being recorded, just so everyone knows, for anyone who wasn't able to make it in person. So if you want to keep your camera off, that's okay. Okay, great. Anthony, if you're ready, you can go ahead and share your screen and get started.
Okay, I just want to start by saying I'm Anthony Odell with the University of Washington Olympic Natural Resources Center and the Olympic Region Harmful Algal Bloom Monitoring Program, ORHAB. I've been with 25 years, and I've been putting on phytoplankton taxonomy, harmful algal bloom taxonomy training courses since about 2008. In 2008, I attended the IOC, the Intergovernmental Ocean Commission's course on harmful microalgae in Denmark and got certified there. In 2019, I took the US HAB course on harmful algae identification in marine waters and got certified there. And then I just recently returned from Naples, Italy, where I went to the advanced phytoplankton course, the 13th one that they've had on taxonomy and systematics. And they started in 1974. So this course only happens about every 4 or 5 years. And it's pretty select. I'm a pretty small group of people that got this training, and I'm really honored and happy that I get to pass some of this on to you over the next few months. I recognize several names in the group already, but just really quickly, if you could kind of give me an idea who my audience is. If you could raise your hand if you have like very little microscope experience. Okay, okay. And how can we get people to raise their hand if they have some microscope experience? Okay, all right, okay. So I'm going to assume that people have had some, been on a microscope a little bit, have been able to look at phytoplankton samples. When you first start phytoplankton taxonomy training, it can be really overwhelming. You're learning a whole other language when you're dealing with morphology, how they're shaped. I'm going to kind of go over morphological terms. There's a few slides that are kind of like phytoplankton 102 or 201 that look at the ultrastructure. You can't really always see, but it's really to make a point. I'm not going to linger too long on those slides, but we're going to cover just basic diatom anatomy, basic dinoflagellate anatomy, and basic flagellate anatomy. And it's probably not going to be a long talk. I will try to go slow because I know there's not a lot of people that have heard these terms a lot. So after a while, they don't really sink in. And when they will probably start sinking in is when we do talks down the line, and we're going through some of the regional species, and I'm talking about the different body parts, and so this is kind of an intro to those terms. And I'm going to share my screen now. There, can you all see that?
Yes.
Okay. And I lost my screen. Okay. So these are kind of a list of online resources that you can use, and you can write them down, or we can share them again later. This talk is recorded, and you can go back and look as well. when you think you have found something and you have identified it using a taxonomic guide, one of the first things that I would do would be go to that first website, algaebase.org. Algaebase, for the most part, say over 99% of the time, they have the most up-to-date terminology for whatever species you're looking at. Sometimes when you use guides that even from a couple years ago, the names can already be outdated. Phytoplankton used to be kind of broken up or lumped together based on morphological features, so how it was shaped. As metabarcoding and DNA have gotten involved in taxonomy, we realize that even though things look alike, they might not be related, and so names have changed. Michelle and I have actually worked to update our species. We came back from Naples with a long list of name changes, and that's just how it goes in the phytoplankton world and taxonomy. The last book on diatoms that didn't have DNA was in, I think, about 1990. And that's still my go-to, but when I'm going through and I'm looking for something, I automatically, after I find it, I will put that name into the Algaebase search, and it'll give me an updated name if there is one, but it also tell me if it's current or not. So Algaebase is really important. Marinespecies.org is another page like this, but it's dedicated to all marine species. But again, you can go and you can put in a whatever name that you find in your guide to see if it's been updated or not. Most of the time, it'll just direct you to Algaebase. Diatom base is really, well, those next three there are really good for diatoms in general, both saltwater and freshwater. Benthic, bottom of the ocean, epiphytic, ones that stick on things or in soils or in Arctic sea ice. So it kind of has like a larger kind of database of diatoms, but it also kind of goes over diatom morphology, their sexual reproduction, or their, how they divide asexual reproduction, and their life cycles and how they're built and things, if you want more detail there. The next one, dinophyta.org, is the same thing for dinoflagellates. It talks everything you need to know about dinoflagellates, and it also has identification keys. So it helps you break down, like it's shaped like this, it looks like this, and it'll help you kind of narrow into something if you come across an unknown or something you don't know how to identify very well. The next several are links to taxonomy guides, and there's some pretty good ones in there. So I will leave it at that. Okay, so these are diatoms. So these are line drawings by a gentleman, Ernst Haeckel, in the late 1800s. And this is kind of when microscopy was taking off in the 1800s. These are all line drawings. So he did this by hand. I don't know about you, but I can't even draw a circle, like a perfect circle. So he did all of this by hand. Very tedious. Definitely took his time. Really beautiful. Like these could hang on your wall. The reason why I'm sharing this is when you're on the microscope, keep a notebook that is just meant for drawing, and just if you can just like draw the outline of what you see and any kind of features on it, it will help you learn. It's one of the best ways to learn on a light microscope is by doing line drawings. There's a full guide on diatoms by Esther Cupp that was published in 1949, I think down in San Francisco. The whole book is huge, but it's all line drawings of diatoms, and that's really how you're going to see and understand them, because it gives you kind of an outline, and it'll show you different views, but it'll show you all kind of the main structures. So if you see anything beautiful or interesting, but especially an unknown, something you've never come across before, get out a notebook, get a pencil or a pen or whatever, and draw a nice little picture of it. For me, I know that helped early on, and I still do it. So when I come across something that I don't know what it is, I will make a little line drawing picture. And it also helps you remember kind of the key features of what you're looking at. Okay, We're going to start with diatoms. For the most part, the most simple thing to identify on a microscope, I find. There is a lot of kind of morphological overlap in between genuses. Like a lot of them look very, very similar, and you actually need scanning electron microscope to tell some of these diatoms apart. Don't worry about that. We're using light microscopes. And if you come across something that it looks similar, you can find the name, at least the genus that you think it might be, and put a little CF in front of it. It means confera, it means for reference. So put CF period, whatever you think it is. So the word diatom, it's a Greek word, dia meaning two, and tomos meaning cut. So it's cut twice, cut in two. It's two halves. It's the largest algal group. As of last fall, there were 16,882 described and accepted species of diatoms. So just that number alone, it puts all animals to shame. It's like, 4 times as many animals in the world, or 4 times as many diatoms in the world than there are animals. And that's just described. Many remain undescribed. I know several researchers that, like, the first time they went and looked in a glacier, they found over 200 unidentified, undescribed diatoms in one core sample. So it's just a matter of going and looking. And so there has been most folks think that there's up to 100,000 species, but there's some estimates over 200,000 species, because people are arguing that, we don't know everything that's in mud. There's a lot of places in the world, kind of tundra regions, for instance, that are just now being exposed. We're finding new diatoms in benthic stuff that's at the very bottom that no one's been down to look. Glacial habitats, like I said, the first time we go and look in glaciers, we find all of these unknown diatoms. So that estimate it's an estimate. It's like the best we can guess. There are between 1,400 and 1,800 marine diatoms, and and the reason why that number is kind of broad is there's a lot of genetic work still being done, and SEM, to determine. A lot of these were described using a light microscope, and you kind of have to find them again and then culture them, and then do the kind of advanced looking with DNA or SEM to determine if they really are a new species or not. Diatoms are autotrophic. This is really important. So autotrophic means they make their own food. So they have chloroplasts, so they photosynthesize. So they're taking sunlight, nutrients out of the water, carbon dioxide, and they're turning it into oxygen and complex carbohydrates like lipids, starches, sugars. And so in photosynthesis, they're making almost about 50% of the oxygen that we breathe. So every other breath that you take is from phytoplankton. So you can thank diatoms for that. Also, it's not just trees and bushes like we're kind of growing up thinking about. Nobody thinks that the ocean is giving us air to breathe, but it is. And diatoms are also responsible for about 40 to 45% of the ocean's primary production. So everything in the ocean, eats phytoplankton or eats something that eats phytoplankton, right? But the very bottom of the food web is diatoms and cyanobacteria, and they're the ones that are really creating, this primary production, and they're the base of the food web in the ocean. And that makes them 20% of the global primary production. So, they're really important for all life on the planet is the point. They're in all environments, all habitats. They just need to be wet and have daylight, some kind of light source. They're in marine, freshwater, hot springs, temporarily damp soil, sea ice, and appear at all latitudes. The temporarily damp soil is actually one of my personal favorites. I raised my 4 children out in a bog, and every winter, our front yard would turn into a lake, a pond, and it would come alive. And the kids are like, what's the smell in the front yard? Like what is that? And why is it that color? And I would be like, here, it's phytoplankton. And be like, Dad, like, you have phytoplankton on the brain. It's not phytoplankton. It's like mud or it's goo or something. Like, what do you think that is? Like, here's the net, go take a sample, let's go look on the microscope. And sure enough, we had all these diatoms and small little flagellates that lived in our front yard that they would just go into encystment during the summer months, and they would just wait for the soil to, have a bunch of water again, and they'd come back up again. So, and you see this even in urban environments, in streets and mud puddles, they'll come alive. So these things can really persist for a long time, kind of in a resting stage. The lifestyle, they're planktonic, which means just free floating. They're tychopelagic, meaning they need a substrate for reproduction. They just need like a surface to touch so they can reproduce. And there are also ones that are benthic, that live on surfaces or on sediment. And so, and even like kind of suspended sediment, they can grow on that. They just need something to stick to. They vary in size from 5 micrometers, which is really, really small. You'd barely be able to see it on a light microscope, up to 2 millimeters. Diatoms are eukaryotic, so which means that they have a cell around the membrane of the nucleus. They're unicellular, so these are single-celled organisms. Even though many times you'll see them form colonies, and it'll look like they'll have radial shapes, or they'll be in these long chains, or like a row of beads. Each one of those little row of beads or individual cells can live on its own, and they are photosynthetic, as I said. There are less than 10 species that have been found to be heterotrophic, meaning they can't make their own food, which means they need to eat something. Usually that's bacteria. They have an exoskeleton, so a structure on the outside that is called a frustule. And it's a cell, or it's a cell wall that's composed of silica. So they're getting silica, they're pulling it out of the water. And it's glass. They live in a glass house. This is a petri dish, right? This is basically what a frustule is. And then there's a link to the diatom.org glossary as well, if you're interested.
Hey, Anthony.
Yes.
Before you move on, there was a question. Could you just clarify for some of those primary production statistics, like what portion you're attributing to diatoms versus all phytoplankton, just for clarification? So there are a lot of different types of phytoplankton besides diatoms, but diatoms are the largest group.
Yes.
But just clarify what those stats are specifically for diatoms versus all phytoplankton.
You mean primary production?
Yes. Yeah.
Okay. So yeah, no, that's most of the other phytoplankton out there are dinoflagellates and other flagellates. They tend to be heterotrophic or mixotrophic, meaning they eat the diatoms. So there a lot of those other species are kind of the very next layer up in the food web. And so really, and the other part that's responsible for the primary production, like I said, is cyanobacteria, which there is a lot of cyanobacteria in the ocean as well. It's very small and we don't really cover it, and you can't really see it on a light microscope, but there's a lot out there. Does that help? Does that answer the question?
Yeah, I guess I think so. Yeah. So just to clarify on your slide where you say 40 to 50% of oxygen is from all phytoplankton, and then diatoms specifically are responsible for 40 to 45% of the ocean's primary production, with the rest of it being cyanobacteria and partially other phytoplankton.
Yeah. So
heterotrophic.
When I say that, okay, the produces 40 to 50% of the oxygen in the atmosphere. So that's all, anything that photosynthesizes diatoms in general, they will make up about 50% of the oxygen that we breathe.
Okay.
When I talk about primary production, that is food. So they're the base of the food web. So those are, even though it's both about being autotrophic and photosynthetic, they relate in 2 different ways. So primary production means food web. So that helps. Does that clarify?
Yes. Yeah. No. Thanks. That's helpful.
Okay.
And send follow-up questions in the chat if you have further questions. But thanks for answering that. Okay. Carry on.
If you want to answer the other questions, yeah, we can do that now.
Oh no, that's all for now.
Okay, that's all for now.
Yeah. Unless there's follow-ups, then I'll let you know.
Okay. Okay. Diatoms, like I said, are composed of a fresh tool. And again, it's basically it's a petri dish. It's composed of a silica wall. And the elements cover the cell entirely, but composite structure allows for growth and cell division. So when there's two cells and there's the epitheca or the epivalve on the top and the hypotheca or the hypovalve on the bottom, and they can come apart so that it can reproduce asexually. The mother valve will form a new daughter inside of it, and then the daughter will form a daughter inside of it. So every time that it reproduces, it gets just marginally smaller. And then when they're down to about 1/3 of their original size, they will produce gametes that will go out to the water and mix, and they'll reproduce with sexual reproduction. The mid area is called the cingulum or girdle. And then you have the hypocingulum is this part of the valve of the hypovalve, and then the epicingulum is the cingulum is like the mid area. It's like the connecting area. On the diagram on the right, we've taken the cell up at the top and we've broken it up into its various parts. You have the valve face, which is the top, and then the mantle is the shoulder region. Kind of think like you mantle over a fireplace. It's kind of, kind of sticks out, or it can, be a little bit beveled. And then you have girdle bands or singular bands. One thing with diatom terminology, which I know can be frustrating, is there's some overlap with dinoflagellate terminology. And if you talk to diatom people that only look at diatoms, they will say epivalve, hypovalve. And then the epitheca is the whole area of the epicingulum and the epivalve. And the hypotheca is the whole area of the hypocingulum and hypovalve. And that's where the kind of the dinoflagellate and the diatom people have kind of come together and say, we agree, this is how we're going to term it. So with centric diatoms, this is the valve face, the top, and this is the girdle along the side. So if you're looking at it from the side, it looks like a rectangle, right? And if you're looking at it from the top, it looks like a circle. So these are three-dimensional objects. So you have to think of them like that. So this, looking at it from the side, this is girdle view, and this is valve view. This is basically a slide kind of covering what we just covered. But I just want to kind of use these diagrams so that you can, kind of see the difference. So this, rectangle view, there's the genus Thalassiocera, for instance. Lots of times you will see it in girdle view because it forms these long chains. Looks like a bunch of wheels. But if you see one that's by itself and it's, laying on its side, and so now you'll look at the valve base. So especially for the first time on the microscope, it'll look like two different things. You're looking at it from different angles. Yeah. So there's two kinds of diatoms. There's centric and pennate, and I'll kind of get into that in a little bit as well. Pennate diatoms are divided into two groups, with a raphe or without a raphe. A raphe is basically, it's a longitudinal slit, and pennate diatoms, if they do have that raphe, there's generally, it's called fibulae. It's like a series of little ribs, and you'll see kind of over here on the right, this is a pseudonychia right here. You can actually see those ribs, so it has a raphe. And the purpose of that raphe depends on, they can use them to attach to each other and form colonies. You can have a keel that'll fit into the raphe, which is like a little ribbed area. So it's like tongue and groove on a board. But they also use it to extrude material to move. So if they, and so lots of times you'll see pseudonychia gliding across your slide when you're on the microscope, and it's actually extruding material out of that raphe. Basically, it's bumping something and it doesn't want to be there, it wants to be suspended. And so it's trying to move away and get resuspended. So basically with diatoms, we have kind of three different versions. It's there's two types, centrics and pennates. And pennates, all of the structure kind of originates from a central longitudinal line. So all the structures kind of radiate from that. It's really easy to see in raphid pennates, that's pennates with a raphe, because the raphe is generally pretty visible because it has all that extra structure and it's wider. But even in araphid pennates, you can still see, if you can see the structure, they have areolation, which are small little dots. They'll all originate from a line rather than from the center out. So when I first started studying phytoplankton myself and was getting trained by Rita Horner in the early days of ORHAB, this was the diagram we were given for pennates and centric, and these were all the terms that we were given to learn. And this was one page, and then we just moved on. So you can see it's quite broken up. I kind of want to make the point with centric, you can see that there's a valve and a girdle view, but this is also true with pennate as well. And sometimes pseudonychia won't have that kind of tapered look. It'll look much square, and that's because you're looking at it from the side. So this would be girdle view, and this would be valve view. So if you can see the raphe like central, then you're probably looking at it in valve view. Any questions so far, Michelle? Heard some ding dongs.
No questions. I just put some information in the chat and somebody joined, but yeah, no other questions at the time. Or well, I guess we should pause and see if there are any questions that maybe folks are in the middle of typing or would rather say out loud. Just give a second here. I don't see or hear anything. Okay, feel free to interrupt later, put it in the chat if anything comes up. Okay, carry on.
Talking about diatoms and the different types and frestual symmetry. So the striae are these little rows of dots, and they're actually little pores, and you can see them. I mean, it's kind of hard in this photo, but I'm going to get into it in a little bit. These pores are either cups, but they can actually go through, but they allow for the transfer of nutrients and waste in and out of the cell. But it's when you see that all points kind of radiate out from this annulus or central point of the cell, and then you know that's centric. And pennates, there'll be like a long line down the middle, and things will go straight out. Now, just because something is kind of shaped roundish, or even more like lime-like, or some are even a triangle, you really have to look to see that striation, those lines of pores and which direction they go, because you can have something that looks like this pennate that'll have the rows of pores will be radiating out from something. So you can't go from just the basic outline or shape to determine if it's centric or pennate. So some terms that we're going to hear down the road is I'm going to say things like the valvar plane, or per valvar axis, which is from like valve to valve axis. The apical plane, that means kind of goes from the top to the bottom. So if you're looking at it this way, the transapical plane, it cuts right across the cell. And this is with pennates. And with centrics, you have the radial planes, which are the ones that go from the outside into the center, and then the valvar plane, which cuts the valve in half into its two pieces. And then up here for diatoms, as I said before, you have a girdle view from the side and a valve view. And you really need the valve view to determine where the striations are, and to see where they're coming from. As you can tell here in girdle view, you can kind of see the two parts of the petri dish, but you can't really see any of the striations to determine if it's centric or pennate.
Anthony, we had another question.
Sure.
which is, can these features be seen with a light microscope without straining?
Yes, yes, and it depends on the genus, of course, but you can see areolation on I mean diatoms and dinoflagellates. You can see those pores. Thalassioseric cocinodiscus are, you can see those lines going, across the valve face very, very well. In some Pseudonychia species, you can actually see the striations, but you can definitely see the rafe and like a central interspace. If you can see that with a light microscope. There are a lot of diatoms actually that you can see this areolation. Not all, not all. So
Would it be fair to say it partially depends on the size of the areolation and the size of the cell, and that is also in itself a clue as to what you're looking at. So for some Pseudonychia, we have like the large ones and the small ones, and the large ones, you might be able to see a little bit more of these structures. And like if you actually can see the striations, that tells you that it's in the larger group. And when you can't see those because it's too small on a light microscope, it's likely that you're looking at a small morphology because it's too small to see. So I guess it's not a blanket rule. Yes, it depends, but for most of the things that we're looking at, or rather for many of the things that we're looking at and identifying, you should be able to see them with light microscopy. I'll also add that hopefully you'll all be able to join either in person or virtually for our meeting or our training on February 18th on Tuesday, where we'll have live organisms to look at under the microscope, and we're hoping to set up a microscope to show folks that are joining virtually or looking at the recordings afterwards to be able to see through the microscope some of these structures on real cells. So we don't have as many light microscopy pictures in this presentation because it's more understanding what the structure looks like with the diagrams and seeing the like SCM pictures, because then you really like know what you're looking at. But we will have more light microscopy footage available in 2 weeks from now.
yeah, that's a really good point. So I've included, there's some light microscopy photos, but a lot of them are scanning electron microscopy, which are very highly detailed, or there's kind of sketches and diagrams. And the point of that is to kind of get you to understand kind of these concepts. And not that you're going to see them on a light microscope, but they may present themselves in a way that will help you understand what you're looking at. So like I said, this is kind of a 101, kind of the basics. And once really, this is what's going to help you kind of get started in phytoplankton taxonomy down the road.
One more question, and can you go back to the last slide, Anthony?
Sure.
Are those two pictures on the left, those are light microscopy pictures, correct?
Yeah.
And so I think also part of it is depending on kind of your settings and your lighting, and also whether or not the cell you're looking at is alive or dead. So if you're looking at a live cell, there's going to be chloroplast and color and different organelles inside the cell. Whereas if you see kind of the frustule by itself, a lot of the areolation is going to be a lot clearer because it doesn't have the like internal organelles kind of blocking the view and the light. So part of it is, it might be a bit harder to see on a live cell, but the other thing you can do is change your focus a little bit. So rather than focusing on the middle of the cell, you're focusing just on the surface. And so there is definitely like ways of changing the lighting and the focus to look for these features, even if they're not immediately apparent.
Yeah, absolutely. And yeah, adjusting your focal plane is like really important to kind of find, especially like the valve face. And the next slides, I'm going to kind of talk about that a little bit. Also, depending on the microscope that you're using, if you have a camera, sometimes I have a nice Zeiss here, and I have an AxioCam 208. And I have Zeiss Zen software, and it allows you to just adjust kind of color, and I can switch things into black and white, or add calcofluor and pull colors out, and you can kind of see different features. So kind of depends on the microscope that you have, but if you have good software and a good camera as well, you can kind of play with it. Kind of bring some of these structures out that maybe you can't see as well just directly into the light microscope.
Unfortunately, a lot of folks in our program do
Yeah.
not have that, but for those of you that do, that's great. And also for the rest of us, we do our best with what we can. And, you know, if we ever get some additional funds, maybe we can invest in some good software and cameras for everyone, because I think it would definitely be super helpful.
It
But
is
yeah,
very cool.
just throwing that out there. Not everyone has that. But yeah, no, it's a good point that if you do, or if especially if not all sites have it, but some sites have kind of better setups, that's the kind of thing where we can kind of connect and share some of those images. Okay, carry on.
Yeah, I think it's really important too that phase contrast when doing Phytoplankton taxonomy or I mean just doing any kind of phytoplankton work, phase contrast is really important. Phase contrast just takes the light and bends it at an angle, and it makes the frustule or the theca kind of like brighten up, and you can see those features well. If you don't have phase contrast, sometimes it can just look like really kind of faded out. And something else that I highly recommend is either a blue filter or kind of a filter that looks like that. And it'll, and I can get you the specs for these as well. But it will also bring out features and bring out colors. Just if you can have just a blue light filter on your microscope, it'll really make the greens and the yellows pop, and you can kind of see the internal structures. And then this lens that I have on here also brings kind of color to things more. Everything kind of looks kind of brighter, especially with things that have cellulose thecas. So. All right, almost an hour, and we're moving on here. So the next few slides, I'm really going to go over fast. They involve kind of ultra structures, and these are things maybe that you can't see, but it was important. It's important for you to know that they are there. So if you want the terminology, I'm going to kind of go through these slides very quick. A midrib, or also called a keel, is like kind of that central, if it's not a rafe, it's on the other side of the cell. If it's, you know, here we have a rafe, longitudinal slit. Those striae are those row of pores, and the interstriae is kind of the hard part in between. And areolae or areola singular are the pores, are those little dots themselves. And so we'll use the term areolation. And that's the design of the areolae on the valve or theca. This again with diatoms, and we are looking at SEM in a diagram here, but with diatoms, the point of this that I want to make is if you're looking at, say you have a centric diatom and you're only looking at one valve. If you're looking at the top, you're seeing one set of areolation, and if you look at the bottom, you might be seeing a different kind of organization of areolation or pores. And so this might be the same species, but if you're looking from the inside, it might look different. So, and that's the whole point of this slide, is it's when you're doing identification and you just have, you know, you're looking at a centric, make sure that you're focusing, you know, your plane of focus is really truly on the surface and not just inside, because it might look a little bit different. It's the whole point of that slide. I'll move on. Yeah, this is again, same thing. This is an example of that. This is Casinodiscus. So this is the outside of the cell, and this is what it's going to look like. And if you're looking at the inside, you're seeing this. And here's a girdle view of that same epivalve. So again, moving on, just to make that point. Again, same thing with the Lasiacera. It looks very different from the inside than it does from the outside. So there's also two terms that you're going to hear, labiate process and strutted process. A labiate process is basically a little kind of protrusion. We used to call it, well, we used to call it a labiate process. Now, we're trying to get to the Latin terminology, rimoportula. Labiate process is an anglicized word. It doesn't translate, and they do phytoplankton taxonomy all around the world. And so we were really trying to get back to using Latin. So rimoportula is what we would call a labiate process. You're going to see that a lot in your textbooks and guides, but it needs to be rimoportula just so it's universal. And you will see this mostly on the valve mantle. You see this a lot in Thalassiocerea. It'll have these little labiate processes. A strutted process is now called a phytoportula. And it is a process that goes all the way through the cell wall. And that's just what these designs are. And here you see Thalassiocerea, it has them throughout, and it's that thread that attaches them, goes through the cell wall into the next cell through the cell wall. And these may function in buoyancy, absorption of nutrients, including metals, and they secrete fibrils. And these labiate processes secrete mucilage. Mucilage, think of it like snot or glue. It helps cells stick together. A lot of diatoms especially secrete mucilage to form their colonies. So they'll have like to form chains to buttress up together, to meet at their feet. And so this is where that comes from, our labiate processes. Okay, move through those real quick. Rita Horner told me early on, I think in the 2000s, that she believed we have about 51 species of Phytophthora in our area. So we know we at least have 51, probably more. Don't get too lost in the weeds when you're trying to go to species. If you can't go to species, it's fine. Phytophthora is enough. Really the harmful ones are easier to identify. And there are several that are easy to identify based on how they form colonies. And so each one of these is its own cell and can live on its own. They are centric, and you are looking at these in girdle view. So they're kind of on the valve face up here. They can be round, but they're generally kind of oblong and come to a point, but they are centric diatoms, and you're looking at them, you're looking at them like this. And so because of that, sometimes you can see that there will be like little indentations or bands, like Catastrophes van Hookerii will have like a little indentation on the side. That's because you're looking at girdle bands. They also have like these long hair-like projections called setae, and they come off the kind of four corners of the side of the cell here, off the ends. And those setae are one of the first things we kind of look at. We look at, first of all, we look at within the cell, are there chloroplasts, the number, shape, size, location? Are they radial? Are they clumped together? Are they central? And the setae, we look at if they have chloroplasts in them or barbs on them. Those are generally the harmful algal bloom species that we see. So we'll be looking for that. They also have specialized setae. So these are the intercalary setae that go up the colony, and then they have terminal setae. And what we look for is where they meet and the juncture type. So are they overlapping? Are they fused for a while and then they split? And where that overlap occurs, does it occur inside the colony, or does it form outside the colony? Is that where they cross? Do they cross again out here? Some of them don't always. Are they sturdy? Are they thin and wispy? Are they broken? Are the intercalary broken off? And then we also look at the terminal setae, how they're shaped. This one, they spread out and then they kind of come back in. Some of them will go straight out. Some of them, curl away. Some of them are sturdy with spines, and some are shaped just like the intercalary setae, but just like pointed in a pervalvular access way. So pervalvular access would be this way. That's through the valves. And then chain shape. So not all of these form straight chains. Some of them are curvy or bent. Some of them kind of form loops that will end up looking like somewhat like a ball. And then sometimes they have spores, oxospores in them. And it's like an extra large lumpy cell. And that is just one of the life cycles of diatoms. And so they'll be forming these oxospores. And you can kind of sometimes use those in certain cases to help you identify at least which species that you're looking at. Okay, in just about at an hour, which that's fair for diatoms, we're going to switch into dinoflagellates and flagellates. Any questions before I move on?
We had several folks ask if we could share the slides afterwards, so they can see all the diagrams and.
Oh yeah, absolutely, absolutely. Yeah, well, we'll get through the talks, and if you want me to go back and look at any slides, as long as you want. Yeah.
Yeah, and also, yes, there is a recording of this presentation. It's being recorded right now, and we will try to process it and share the links of it as soon as possible after today.
Yeah.
Any other questions on diatoms before we move on?
Oh.
Looks good.
Okay. So we're going to move on to dinoflagellates and flagellates. Dinoflagellates, also known as lerbites, little round brown things. People send me pictures all the time. What is this? And it's a lerbit. It's a little round brown thing, and it's because it's fuzzy, it's out of focus, or I can't see the parts of the cell that I need to identify it. So yeah, little round brown thing. You see them a lot. One of the HAB species we look for is alexandrine, which when you're looking at it from the top or bottom is a little round brown thing, right? If you really need to see it kind of from the side to determine what it is, and the ventral side, the front side. So yeah, dinoflagellates are mostly unicellular organisms, about half of which are phototropic. So some of them can, get their energy from sunlight. And it says mostly unicellular because some dinoflagellates will collect into kind of one body, and they will kind of share the material in them, and you call that a zooid. And so they're technically multicellular, but they can live on their own, so they're unicellular. Most or many have now been shown to be mixotrophic, capable of ingesting food in addition to their phototrophic mode of living. Other species are heterotrophic, and they only feed. Many only feed. There are 2,000 living species and about 2,500 fossil species. 1,700 in marine habitats, and 600 to 700 of those are athecate, meaning that they don't have an outer shell, or very light outer shell. There are unarmored, athecate or naked, lightly armored and heavily armored thecates. These are descriptive terms that do not carry taxonomic meaning. And this is really important because you can't lump things into, naked or armored, athecate or thecate, because it's a spectrum. And so the lightly armored to athecate, people argue about that. They're like, I think I kind of see something there. So, and I will kind of get into that, but yeah, those are not taxonomic terms, but you're going to hear them a lot. It's kind of a way to lump dinoflagellates together. The nucleus has permanently condensed chromosomes, which can usually be seen in a light microscope. There's like these little curled striations in the nucleus, and it's called a dinocaryon. And if you can see the nucleus, you can actually see the little lines of chromosomes in there. And yeah, that's how you know that it's the nucleus. And you can see that with a light microscope in most cases. Dinoflagellate cells are covered by an emphysema, which is a complex of vesicles surrounded by the plasma lemma. So they have a skin, basically. And then whether that skin is armored or not, that's something else. So they are also known to have diatom, dictyophyte, and cryptophyte endosymbionts. So over the hundreds of thousands of years, as they've evolved, they have been known to kind of steal other parts or have other things living inside of them, which you can actually tease apart and culture and grow on their own. So these endosymbionts are really dictophytes and cryptophytes and diatoms that live inside of the dinoflagellate. And actually, the course in Naples actually saw some of those that were grown in culture that they don't exist really in the environment, and they when the cell divides, they just like also divide the diatom or the cryptophyte, and it goes out and it joins back into the dinoflagellate. So they don't really exist in nature; they only exist as endosymbionts. And then some species, many species actually, there's a group that have kleptochloroplasts, which means stolen chloroplasts. So Dinophysis will do this, Nematodinium will do this, and so they're heterotrophic or mixotrophic. This is why they become mixotrophic, is they will eat something that has a chloroplast, and rather than consume the chloroplast, they take it and they make it a part of their body. So, rather than eat the chloroplasts, when you're consuming it, think of it like a donut, right? It's lipids, it's starches, it's sugars. Okay, that's what it's produced, so that's what's in there. So rather than eat the donut, they just keep the donut factory and it's part of their body, so they always have donuts. That's a way to think of it.
Anthony, we had a question.
Sure.
Why aren't dinoflagellates that ingest food and move not considered animals?
They're singular. Well, that goes back to genetics and DNA. They're not really remotely close to animals genetically, but also they are single-celled, and they have parts of them which are closer related to plants. The theca, the shell of a lot of these armored ones, is actually cellulose, so that's more plant-like. So, and that's maybe in the taxonomic training coming up, I will kind of show you the tree of life and where animals are and where dinoflagellates and flagellates and diatoms are. Genetically, they're very different. So, yeah, they don't fit into Animalia. We're closer related to fungi, actually, than we are dinoflagellates. So.
And zooplankton, what keeps them all together is that they are multicellular.
Yeah.
In general, so.
Yeah.
All right, thank you.
Yeah, no problem. So we have two types of dinoflagellates, desmocont and dinocont. Desmocont is really, they just have two valves that go around either side. The flagella is usually apical up at the top that stick out, and that's really just kind of prorocentrum types, and it's a pretty small group. And most dinoflagellates and flagellates, most dinoflagellates are dinoconts, which means they have two parts, a top and a bottom, which is either the epitheca if it's armored, or an epicone if it's unarmored, and a hypotheca or hypocone. They have this kind of longitudinal furrow if it's naked, or a sulcus. And it's basically kind of a slit that goes from the bottom up to the middle. Sometimes it can go past or even up to the top. But the important part is there's a flagella in that sulcus they call it a trailing, and it basically pushes the cell along. And then they have what's called a transverse furrow or cingulum or girdle. And in that is the transverse flagella. And this provides, and this kind of wiggles up and down all around the body, and this provides motility, and this helps them to kind of like steer and move. So they have one flagella kind of propels them, and the other one kind of steers them, orients them. Okay, I know there's a lot of letters and numbers on these diagrams. Those letters and numbers are called cophoidinium tabulation, and we're not going to get too much into that because you really need scanning electron microscope to see all these different plates on most dinoflagellates. You can see them on some that are lightly armored, and you can actually see these plates. So don't get too worked up about cophoidinium tabulation. It goes from this is considered left to right. And so the front where you see the sulcus, that's the ventral side. And the back without the sulcus, that's the dorsal side. Anything on top, anything above the cingulum, the central area, is called the epitheca. Anything below it is called the hypotheca. The top is the apex. The bottom is the antepex. Anything near the bottom is antipical. Anything near the top is apical. And then you have pre-singular and post-singular. So pre-singular just means right above the cingulum. Post-singular means just below. Intercalary is that kind of gray area between the apex and the cingulum that's above the pre-singular. I wouldn't worry about intercalary too much. That's really only used in Cophyllodynium tabulation. Here's kind of another same thing over here. Hypotheca down below, epitheca up above, cingulum in the middle, sulcus, apex, antepex, apical ends, or antipical end and apical end. And you can see here again, this is why they're called lurbits. If you're looking at something from the apex or the antepex, it's just going to look like a little round brown thing, right? And you can see there's like kind of a sulcal area here, or a furrow, a little bump that goes in that tells you this is the ventral side and this is the dorsal side, but you really don't have the identifying things that you need to help you identify the dinoflagellate. Ideally, especially if you're sending pictures to Michelle or I to help you figure out what something is, we want to see it like this. We want to see it on a ventral view where we can see the sulcus and the cingulum and any ornamentation on the apex or antepex. It's kind of what we're looking for. So dinoflagellates can be broken up into two groups, paradinales, paradinoids, or gonialicales, gonialicoids. Paradinoids are symmetrical about the sulcus. So if you're looking at the ventral view from the front, all of the plates, everything's kind of arranged in, for the most part, fairly mirrored fashion. And the cingulum wraps around and it comes back to itself. Okay, that's a paradinoid. Gonialicoid, the cingulum is offset. And so it wraps around and it kind of, as it's wrapping around, it drops, called left-hand displacement mostly, where it drops and comes back in underneath where it starts. You can also have something called, this is called overlap, where the cingulum, it doesn't not only not meet, but it extends past the starting point. It stops past the starting point. You can also have something called overhang, where the epitheca and even part of the hypotheca will overhang and kind of cross over. It'll have like a flap that protects the singular flagella. And so again, about the axis, you can see that it is not mirrored. It is asymmetrical. And if you're looking at it from an apical or antipical view, you can see again that the plates are not mirrored. And lots of times you'll see that there's some kind of overhang. It kind of goes beyond, like this is kind of inside and it wraps around to like an outside cover as well. Yeah, there's several different orientations for that, and I will get into that a little bit. So paradinoids and gonialicoids. And if you're describing something to me, it's one of the first things I'm going to ask. Is it paradinoid or gonialicoid? Most dinoflagellates tend to be gonialicoid. So these are all different orientations for the cingulum and sulcus. A is displaced with a sulcal intrusion into the epitheca. So the sulcus generally will stop at the cingulum. Sulcal intrusion into the epitheca is one way to describe a cell. You're going to hear this a lot. It's displaced. Displacement just means that it doesn't come back around and meet itself. It drops down, and that's displacement. And it also kind of, there's a little bit of overshoot, right? It doesn't, it's a little bit longer and goes past it underneath. That's displacement. We measure displacement by singular length. So this would be like probably displacement of 1 or 1.5. singular widths. So displacement of 1 or 1.5 left-hand displacement, what would say. And we talk about where the cingulum is placed on the cell. So you know which end is the bottom, the posterior, because it'll have the trailing flagella. Okay. So if it's above the middle, it's pre-median. If it's in the middle, it's median. If it's bottom, it's post-median. So we also see that there's a large sulcal intrusion into the epitheca or epicone. Same here, there's a small one. So we'll talk about like how far that intrusion goes. Sometimes in some of the naked flagellates, this sulcal intrusion will end up and go into a groove on the top with there's some gymnodinoid species, and the shape of that groove and the sulcal intrusion are ways to determine which genus and species it is. Down here in E, this is what we call overhang. And again, these cell plates on the hypotheca and the epitheca, right around the cingulum, they kind of wrap and cover that trailing flagella where it comes out. So it kind of provides protection for that flagella. And here you have a displacement without singular intrusion into the epitheca. And then this last one down here is called torsion. And you'll see this on a lot, especially of the kind of the more highly evolved dinoflagellates. They'll have a great degree of torsion. So their cingulum and their sulcus, it's like you took the cell and you just twisted it. And what that does is it provides a high degree of motility. So these things can, stop, spin around, turn on a dime. Nematodinium armatum it's like one of my favorite dinoflagellates. It has a high degree of singular and sulcal torsion, and it'll be swimming along and it'll see its food source and it'll like back up and then go after it. And so, yeah, this is it's pretty advanced, but you can see that, this the cingulum like wraps several times around, and the sulcus with it. So it's like the whole cell just got like forked. Okay, and then for atheca dinoflagellates, it's basically all the same, right? Except for now we're not saying epitheca and hypotheca, we're saying epicone and hypocone, but everything else remains the same. With atheca dinoflagellates, though, you can see inside better. So the armor is not, in the way of seeing the organelles inside. And so you can see chloroplast, and you can see trichosis, and you can see nucleus better and things. So you can actually see the inside better of naked flagellates. So those will help. Those inner parts will help you identify where the nucleus is placed within the cell. You can use with atheca dinoflagellates, how the chloroplasts are arranged, kind of where they're at, their shape, etc. But everything else is the same. Still have the sulcus and the cingulum, apex, antepex, longitudinal flagellum, the singular flagellum. They're all, everything else is the same. So just basically the only difference is instead of saying epitheca, this doesn't have a theca, so it's epicone. Instead of hypotheca, it's hypocone. Now, this is my last slide, and these are very small flagellates, or other, aka URSOs, unidentified small round objects. These are those little zoomers, as Rita would call them. There's like a little ball that goes by really fast. They're very, very small. They're very hard to identify with a light microscope. Sometimes you are able, especially if there's a bunch of them. If you preserve them, they just kind of shrivel, and the flagella or haptonemas, things will fall off. And so they will just look like an unidentified small round objects. And that's kind of the problem with looking at preserved samples. If you put Lugol's on them, a lot of them will shrivel and shrink. Sometimes you can identify with a light microscope after that, but especially with the very small ones, again, it just looks like a little brown dot. So the important point that I want to make is you really need to be looking at live samples if you have the time. So and as soon as you get back from sampling, you should try to get on the microscope right away, because a lot of these things won't live overnight. If you want to keep it overnight, try to keep it as close to the water temperatures you took it from. And so, not necessarily put it in the fridge, but keep it someplace cool or warm if that's the case. take the lid off, crack the lid at least, give it some air. There needs to be gas exchange if you're going to look at it a little bit later, but you do need to look at live samples to find these things, really to identify them. A lot of them just don't preserve. Some of the larger ones you can, but most of them are very, very small. So one of the first things that you would do if you saw something swimming by, you look at the flagella, see the number, smooth or scaly. So most of them are smooth. Sometimes you'll see like little bumps or ridges down one side, and you know, it's gonna have to sit still, and you're gonna have to be like looking at least 40x or closer, or 75x oil immersion to actually see that smooth or scaly. Sometimes you can see it with just like as they're swimming. Length equal or unequal. So we have two flagella. We have one going forward and one trailing. Sometimes you will have, one and they'll have like the same length. One will be longer than the other. Kind of also the flagella, the origination point. Do they come out of a furrow or like a little gap, or is it set up like a naked flagellate where you have a cingulum and a sulcus? Most of these, they have several flagella coming out of the apex, and you can have up to 8 in cases. And naked flagellates, these very small flagellates, out of the 3-week course in Naples, which is the Advanced Phytoplankton Taxonomy course, we spent a whole week on this, and I'm giving you one slide because with a light microscope, they're so difficult to identify. I think most people are looking at preserved samples and not live samples. They're very difficult to take photographs of. And so when people send me pictures of them, it's like, I can't help you with that. If you send me a video, I might be able to help you with that, because the swimming behavior and their motility often will give away, at least to genus, what it is. And I have several movies as well of all these things as well. I don't know how much time I want to spend on it because they are small, but of all the groups of phytoplankton, as you can see, there's a huge amount of, this covers a lot. There's a lot of diversity in small naked flagellates, but they're so small that, what you see in taxonomy lists is unidentified small flagellate or unidentified naked flagellate. And this like the biggest group in taxonomic write-ups when people are doing taxonomic work, because they're just too small, too quick, and they don't preserve well. So if you see it on a live sample, and there's a bunch of them, especially look for cell shape. Unfortunately, like one thing we've been seeing a lot of, like a primesium out here, is very small. Unfortunately, for a lot of these, there is a high degree of morphological variability, which is my least favorite taxonomic term. It means their body shape isn't always the same, and it changes a lot. Some of these are really amorphous too, and their gut content determines their cell shape, and so they can just be blobs. Some of these can change their body shape, as in this kind of like they use it as a form of motility, and so you can't really go on morphology. You have to look at swimming behavior and cell shape and flagella. So also some of these very small flagellates, they can have skeletons, exostructures. Dictyocha is one of those. That's a silica exostructure, but it's a small little haptophyte that lives in there. Cell covering, wall scales, no covering. Again, this is something, if you can get one to sit still or it has died on your slide and you can zoom in, before you preserve it, you can actually zoom in and look. You might be able to see if it has a wall scales or covering. Chloroplasts or accessory pigments. So the color of these will help you a lot. If they're bright green or red or brown or orange or yellow, that can actually, if you're using taxonomic guides, the color and pigments, that will help you a lot, kind of guide and figure out what you're looking at. The presence or absence of a haptonema. A haptonema is a feeding tube that exists kind of between flagella on the apex, and it comes in all kinds of different shapes. It can be very long, it can be very short, it can be stiff and unmoving, it can be coiled, it can kind of work like a feeding tube and kind of go back and forth. look, it's the third flagella, okay, but it's not a flagella, it's a feeding tube. It's the haptonema. We have lots of haptophytes around here. And that's, it's a good way to group one of these, like you can head towards, at least it's a haptophyte and go from there. Furrow or grooves, and again, that's sometimes you can see these cutouts where the flagella come from. Here's again, because this is athecate, but you can see that it has a groove. This would be the furrow. An eye spot or sigma. So you see these little black dots here. They do have kind of rudimentary eyes that can detect light, and it helps them with vertical migration. So they'll move towards the light during the day and they can go up and feed, and then they'll drop down at night. And then some of these will have what are called a lorica, which is made up of different things, but it's basically like a covering that they live inside, and these will form kind of colonies that way, but you can see them free swimming. Sometimes they'll be in it, and then sometimes they'll be in the lorica. So yeah, sometimes you can see two different forms of them. Again, out of a 3-week course, we spent a week on this, and I'm giving you a slide. They gave us this at the end of the course when our brain was already full as well. And we just kind of like, okay, now we're going to do all of the other stuff. There's a lot of other stuff that is highly diverse genetically. But, we do have several very small flagellates. And we do have, euglenoids and haptophytes, and we have Primesium polylepsis now, which is it's a massive fish killer. These guys are around, and these kind of the basic stuff to get you going on that. But if you're doing phytoplankton taxonomy, there's a reason why this is always unknown naked flagellate or unknown small flagellate. They're very hard. It's probably the last you're going to learn. You'll probably learn diatoms first and a few dinoflagellates. You'll get into dinoflagellates, and then if you kind of become intermediate or next level, then you'll start getting into very small flagellates or ursos. So that is slide number 22 of 22. We're an hour and a half in, so we have an hour for questions, or for me to go back over slides that you'd like to look at again.
Thanks, Anthony. One quick comment just on that last bit is for the purposes of our program, I'm specifically speaking the SoundToxins here, because our primary purpose is looking for HABs, the most important ones for us to identify are the ones that we've identified as target species in our area. So as of now, the most important small flagellates to look for are Heterosigma and Phaeocystus. So we have specific ways of identifying those two specific ones. Now, Anthony mentioned a couple of others, including Primesium polylepsis, which they've been seeing on the coast. And anything you can see in the coast, could come into the, Strait of Juan de Fuca or the Puget Sound at any time. And a lot of these, even though they sometimes have a preferential area to live in, like coastal, like some prefer more energetic coastal waters, some prefer, lower salinity estuarine waters, but they really are very adaptable and unique. And so even some of those old rules where, if you read taxonomy books and it says only found in, super saline environments, those are not necessarily hard rules anymore. And I think these organisms can be very adaptable. So not to say we might see other harmful species come in to our areas that we're monitoring. And so it's good to be aware of other ones. And it's especially important to, when you're looking at your samples, apologies, the abundance is important, right? If you're just seeing one or two and you're like, what's this weird thing? Our curiosity is good and important to follow up with. But if you're seeing a lot of them, that's when it starts to be a question. Or if you see a couple one week and you see a couple more the next week, and you're saying uh-oh, this looks like it's growing into more of a problem, that's where we'd want to try a lot harder to, identify them. And this is also the reason why we do whole water samples in addition to our net toes. because if there's anything super tiny that kind of goes through the net, you'd be able to see that in the whole water sample. So just checking those and seeing if you're seeing kind of like a lot of something, if you're starting to see, color changes and things in the water, that's where we would try a little bit harder to identify these really small flagellates. And other than that, for the purposes of our program, it's not as important for you to be able to identify all of them.
Yeah, and to that point, late last fall, I sampled out at Claylock here, which is kind of north central coast, out on the coast, and I saw an amoeba, and it's single cell, but it has like these weird arms, that kind of foot poles that kind of go out and it reaches, and it's really amorphous and moving around. And I've always thought they're interested, but I've always just been like amoeba in my phytoplankton. Well, I started seeing a whole bunch of them. I'm like, I should probably figure out what this is. It turns out it's a genus that has been responsible for urchin die-offs in Nova Scotia. And so it was a HAB species, but it has to be in abundance, right? But I'm glad that there was enough of them. I'm like, I should probably figure out what this is. So now there's, one more HAB species that we have out here. But I'd never, if it hadn't bloomed, I wouldn't have worried about it. So yeah, to that point, start seeing a bunch of something, you probably want to take a look.
I actually was going to ask you about this because I believe some folks, some of our monitors saw some amoebas recently. I don't know if it's just one or multiple. I should probably follow up on that now that you reminded me that they could be HABs. That's good to know about.
I got asked the same question up in BC, and they want to know what my expertise on amoebas, and I'm like, an amoeba. I haven't, they're very few. They're interesting, but they're very rare, and so I never had a problem. They're like, we've been seeing blooms of it. I'm like, oh, and then yeah, sure enough, we had a bloom later on the next month. So
yeah, good to pay attention to. Okay, so now we have tons of time for questions, and I have some questions that I can ask if no one else has them. One person asked if you could just go back to maybe a diagram of dinoflagellates and like kind of re-explain like the difference between the cingulum and the sulcus.
this one,
Yeah, I think
okay,
that's a good one.
yeah, the cingulum is like, it's like this band that goes around the middle. It's like the belt, okay, and it's kind of excavated, or it's cut out, and that gives room for a flagella to wrap all the way around, which it starts at roughly the same area as the trailing flagella, but this transverse flagella sits in that groove around the outside of the dinoflagellate, and it wiggles back and forth, and it allows the flagellate to move. The sulcus is on the belly side, and it's generally from kind of the belt down, and it's a furrow or a groove that goes down that has a transverse or trailing flagella, and that pushes it. So it has a flagella that sits in that. Now you can't always see the flagella, but you can almost always see the sulcus and the cingulum. You can almost always see that those grooves that exist, even if you can't see the flagella going back and forth. If you're zoomed in enough, you can see them if they're still there. They also break off very, very easily as well. But yeah, does that help a little bit?
Yeah, I think so. And I'll just add a little bit of nuance to that. The sulcus and the cingulum are features that are on most dinoflagellates, but they look different sometimes, right? So that other, maybe you can flip the slide a couple more where you show all the overhangs and displacements.
Yeah.
I was gonna say, I think, so there are some phytoplankton that have like really wide sulcuses, or cingulums, sorry, and some that have really narrow ones, and then some that have, especially the like armored ones, when they have really thick armoring and plates, sometimes they they like stick out. There's like
Yeah.
a bit that sticks out around the cingulum, and we call that a list sometimes. And a good example of this that you might know is I think so. One of our important HAB species is Dinophysis, and that one's a pretty unique one because it is a dinocont, but it's got kind of like in picture B here, the cingulum is really, really close to the top of the cell. So you almost sometimes can't even see the top of the cell. Actually, most, yeah, I think in all Dinophyses, you can't see it because the lists or the parts that stick out that are like protecting the flagella kind of make kind of like a crown, right? So
And then it'll also have a sulcal list, which is on it looks like a dorsal fin, but it's on the front. So it's a ventral fin. And that list, or that big kind of fan that sticks out, also protects the trailing flagella as well.
Yeah, yeah, we sometimes call them wings, and they even have like kind of like the structures that kind of hold them up. So yeah, Dinophysis, it's a really unique one because we were always talking about wanting to see things from the dorsal side.
Mhm.
But Dinophysis often, because they're so, what they say, what, dorsoventrally flattened, so they're really flat
Mhm.
cells that they tend to fall on their sides
Very honest side.
on a microscope slide. And because of that, we're usually seeing them from the side view, and it's very rare, and they actually kind of look disconcerting when you see them from the front. Not disconcerting, they look different than you expect from the front. But for most of the dinoflagellates that you'll see, the ventral side is what you're looking for.
There's also something called reticulations. And reticulations are where you have it like a list, where it's like a little raised kind of like fan. Reticulations will generally go along the cell plate wall. You can see my pointer there. So it's like reticulations would be like a little raised little wall coming up kind of straight out at you. And that you see that like in Protoceration reticulatum, for instance, or have like little raised little lines, little walls coming off the theca, or the outer shell.
We also had a question of saying line drawings are a lot easier to identify, but what is observable in microscopy, and at what magnification would you be able to see these? And I'll just say that we ran a little bit out of time because we wanted to add some more microscopy pictures to this just to show the both sides, but we communicated that a little bit late to Anthony, and so we didn't have time to add them in. But again, we're gonna be seeing a lot of live cells on microscopes on our in-person day, and again, we're gonna try our best to have like a clear picture come through Zoom. So we're gonna try to test that out hopefully beforehand to make sure it works. But we're hoping you'll be able to see this on Zoom. But Anthony, maybe you can talk a little bit about like what magnification you use, and kind of just a little bit more information about that.
Well, I will generally, I have a 10x objective, and I will when I'm looking at a live sample, I will just kind of look over to get an idea of relative abundance, thing that's out there, what's dominant, prominent, and then I will switch down to 20x to do like if I'm doing cell counts or something. But if I come across something I want to do a line drawing of, I will go to at least 40x, or I have a 75x oil immersion as well. And then you can get close enough where you can actually sketch the detail of the cell. And again, that I mean, even if it doesn't matter where you're at, if you want to draw a colony of something, you're just like, I don't know what this is, or you're vaguely familiar with it, and you just draw a picture of it. It's just a learning tool. It's a way to help you remember, because when you're drawing it, you're looking at the identifying features. You're drawing the identifying features. So the things that you can see, you're going to make little line drawings, just like a quick sketch of it. And that's how you're going to remember it next time. So these are single celled organisms, and they're very complex. They can be simplified. Most of them can be simplified into a line drawing, and it helps you remember.
I'll also share one more thing. So often when I do these like intro to phytoplankton things, I'll go through all of the terminology, and then I spend the like last hour just like rapid firing going through like 70 species with pictures. And I don't know, I think it's useful to get an idea of what you're seeing and seeing a lot of different things, but I imagine it's quite hard to absorb. It took me a really long time to learn, phytoplankton, and so I feel like that kind of can be a lot. But one thing we were thinking of doing and are planning to do in this year is we often do like kind of deep dives on how to identify HAB species, because they're the most important for the purposes of this program, to be able to identify and look for. But because the majority of what you're seeing in your samples are non-harmful species, and that's also what can be quite interesting and fun to understand, like the diversity of the ecosystem and what we're looking at, we also want to be able to identify those. And so we're planning on doing a series of webinars where we kind of go through in a little bit more detail on groups of non-toxic non-toxic and non-harmful phytoplankton, like the good ones that you see in the water that are like, good, healthy food for the ecosystem. I'll spend some time on diatoms that are often neglected when we talk about HABs. We only talk about Pseudonychia and nothing else. So especially for those ones that are all very similar and harder to identify, like what to look for. Like for me, it's the kind of chain forming cylindrical ones that kind of all connect into like long slim chains. They don't have spines, so it's harder to identify those. Those are the ones that are trickiest for me. So we're hoping to go through those kind of maybe like one webinar a month, kind of a group of species, maybe like all the Chaetocereruses by species, maybe all the chain forming ones, maybe
And then also,
Cosmodiscus and Bacillosira in one go or something like that.
yeah, we'll go through kind of grouping things morphologically. So things that would be difficult to identify based on their shape, or how they form colonies. We'll go kind of group by group the things that are tricky to tell apart.
So this was just kind of like an intro to terminology in preparation for our in-person meeting, and then throughout this year, we're hoping to have more virtual webinars, which we've gotten a lot of interest from folks about this, and I personally am like really excited about it. So we're hoping to get that going this year.
Any other questions or slides that you'd like to look at?
I'll give those a couple seconds. I do have a couple other thoughts to throw out for the group. So I think the question or thought that I wanted to throw out is that we're covering today. You covered kind of the main groups of what we're most looking for and most often seeing, which are diatoms, dinoflagellates, and then all the tiny small flagellates that you might see zooming around. But there's a lot more that we might actually accidentally catch in our nets. Could you, and we mentioned amoebas briefly, but could you talk a little bit about ciliates?
About what?
Ciliates?
Oh, ciliates.
Yeah.
Okay.
I know you don't have slides for it, but I thought maybe you just
No.
make sure we're covering everything.
Ciliates are I mean, multicellular. They're considered a zooplankton, but they're very, very small. They're on a phytoplankton scale, and a lot of them, like tentinid, will like live in a lorca or a shell, and you'll see like a little animal come out, and they have like little, many little cilia going out that they use to feed with. Another one that we have is Mesodynium rubrum is a ciliate, and it looks like a little light bulb, a little red light bulb with little kind of cilia off the side, and it kind of jumps around really quick. Something else to do when you're doing taxonomy. If you see something you don't know what it is, give it a name, even if it's the wrong name. And Rita Horner taught me this, and what made me think of this is Mesodynium rubrum, this small little ciliate. She named it Space Coop. She didn't know what it was, this little red thing, but to her it looked like a little spaceship, like a coupe that you would drive around. So she called it Space Coop until then she crossed it out. And now it's Mesodynium rubrum. Mesodynium rubrum, although it's a ciliate, is also food for Dinophysis. So it's small enough that a dinoflagellate will consume it. And yeah, there's a few that are common around here: Helicostomella, Favella, Tintinna, Tintinnis, that are all grazers. Both ciliate and zooplankton grazing on certain HAB species can cause them to produce toxin. We know this is the case with Pseudonychia, that it can grazing by ciliates and zooplankton. So you see those in the water, and they're grazing on the Pseudonychia, can kind of stress the cell, and they will produce the toxin. So I mean, it's good to at least know what they are, even if you can't name them.
Yeah, and cilia are very similar to flagella, but also very different. They're usually shorter and more numerous. So if you see things that look like hairy, and the hairs are moving, that is likely a ciliate, not a kind of in the phytoplankton group of dinoflagellates or flagellates. Although small flagellates can have, 4 to 16 flagella. So more than 16 flagella might be
Yeah,
ciliates.
yeah.
Cilia. And then Mesodynium rubrum is one that we have asked folks to look out for, as Anthony said, because it's a primary prey species of Dinophysis. So if you see a lot of Mesodynium rubrum, which is called that because they can bloom and they can turn the water red, because they are reddish colored cells. And if you see those and have some Dinophysis in the water, it could lead to a Dinophysis bloom. So good to look out for. And then, yeah, the other ones that we frequently see are Tintinnids, Tyrrhina,
Yeah,
a
yeah.
couple of other things that'll like zoom around quickly. But yeah, I guess looking for the cilia versus flagella is the way to differentiate those two. Paulette asked that some folks have lots of unidentified pictures saved, and was wondering if we could do like a webinar to go through those to see if we can identify some of the ones that they see often but don't know what they are. That seems like it could be fun. What do you think,
Yeah.
Anthony?
Yeah, I think it would be a very long webinar because I think there's a lot of people with photos. We were actually kind of talking about this. I think the last SoundToxin meeting is creating an online database. So I get photos of unknowns all the time. And kind of one of the reasons why we have this Phytoplankton 101 is so that if you come across an unknown, now you have an idea of the identifying features that I would need to help you identify it. The dinoflagellates, like I said, we want to see it from the front, the ventral side, so I can see, the cingulum. It has offset, the sulcus, any identifying features. If you're looking at it from the apex or the antepex, it's a little round brown thing. We can go through pictures, but there would be a lot that'll be just like, I don't know. So I get that a lot. And then even when I was in the course in Naples and my instructors were like literally the best in the world, and several came out of retirement to teach the course. And I would get photos from folks in this group, in my group in Oregon, and they would send me like, what is this? I know you're with all the experts. Can they tell me what it is? And they would look at the picture on my phone and they'd be like, oh, wow, that's really interesting. So there's a whole bunch of stuff out there that, even though I'm an expert, it might not even be in literature. There's a lot of undescribed species. And if it's like, the first time you've seen it, which I have a whole list of unidentifieds. I'm currently in possession of Rita Horner's her camera photo slide collection, and I'm digitizing it. And she has this broken up. The boxes are broken up into different categories, but she has several pages of slides of unknowns. So things that Rita Horner in her entire 45, 50-year career could not identify. So sometimes they're just unknown. But I mean, I'm always fascinated, and please, you can. I mean, I love sleuthing for unknowns. If I can help you, I will. Sometimes it can take a long time looking through the books to figure it out. It's not. I don't have all of this stuff off the top of my head. I spend a good portion of my time in taxonomic guides. So, but you can send me pictures if you want, and then maybe the ones I could identify, we could go over. Or also, if you come to the SoundToxins course, if you have photos, you can show me there, and I can help you. But yeah, I spent a lot of time trying to answer those questions. I don't know how long the Zoom training it would be.
I was gonna say we could do some sort of like office hours where we have a few and we go through them quickly and see if there's any we can identify quickly, and the ones that might take a bit longer we can like kind of save and return back to them or something. Because sometimes it is the kind of thing where you look at it and then you're like, okay, I have to like think, I have to go do some research, and then I'm like, oh yeah, that reminds me of one that I saw, and then it comes back to you. But there are probably some that a more trained eye that has a lot of experience might be able to be like, oh, I see immediately this thing that you might have missed. So we can definitely kind of set something like that up. And then someone else asked, is it that there are just too many variations that a key is not possible? They've been looking for a dichotomous key, but it does not seem like they exist. And I think that is that you've kind of answered your own question there. It is hard to just have a dichotomous key because there are so many. I think maybe sharing that picture of that tree of life, Anthony, could be useful just because, you know, even based on, where am I going with this? Based on morphology, there's multiple. It's hard to do just like a dichotomous key that just kind of like breaks things down because there's just so much variety. And also a lot of them are not related. So you have things that are kind of like related genetically and things that are completely different but look the same. And so it is quite hard to get a key that you can just kind of follow to answer things, unfortunately. There are in some books, like so in the there's a identification book by several folks, but we call it the Tomas book because it was edited by Tomas. Anyways, within that book, there are a few like genus specific, like to get down to the species level of looking at like kind of like a dichotomous key of saying like, okay, so you were looking at Chaetoceros and you're trying to figure out what species it is, you can follow this key to figure out what species it is. Unfortunately, that one can be a little frustrating because a lot of the things that are differentiating the species at that level are ultrastructure things that you can't see on a light microscope. So I haven't even been able to use those really. But again, you can't have one for like all phytoplankton. I think that would be quite hard to do. You can have some for kind of like smaller groups if you're like down to like it could be this or this, follow a key to figure out the specific species, but I don't think there's one that exists for everything.
I'm looking for a tree of life. One. Okay. I'll share my screen again. It's actually right this time. Okay. All right, you can all see that. So this is like one of the original Tree of Life sketches by Ernst Haeckel. And he's the guy that did that really beautiful line drawing earlier on. And this is kind of how he had things broken up. So this is like one of the first attempts at, taxonomy, really breaking things up and describing them. And now we're moving on. This is basically what yeah, land plants, red algae is in there. Here's land plants, chlorophyta. Um, here's fungi down here. All of these kind of these others that we just covered, they are really throughout all of this tree of life. Okay. Yeah. So that, I mean, this is kind of the last version. There there exists several different versions of the tree of life right now because people are still doing the DNA work and realizing that there's some of these are broken up differently than we assumed that they were originally. Um, just, yeah, that's one. Once
We'll
again,
say there, just to answer your other comment, Doug, there are some high tech machines now, like flow, IFCB. What's the I stand for? Flow cytobots.
yeah, yeah.
Sorry, I forget
Internal. I
what is it?
think it's internal.
Internal flow
It's not about, yeah, it's a
cytobot.
capillary tube that goes through, yeah, in front of the.
And basically, there's yeah, technology now where you can drop one of these machines in the water or inject water into a machine. It puts it through a teeny tiny tube and pushes it in front of a camera one cell at a time. They take pictures of it, and people are also doing a lot of machine learning to then categorize those pictures and identify them. And so those are really wonderful. We have a couple in the area that are used to identify phytoplankton in really quickly in a couple of different areas. The problem with those machines is that they are very expensive and take a lot of maintenance. So even if we had the money to buy one, you need like a full-time person kind of running it. But we do have a couple that are getting really like high quality phytoplankton data in some areas in Puget Sound. But the value of human taxonomy and doing the microscopy is that we're able to get so many more sites and so much more frequently. So that's why this program is wonderful. And we thank all of you for sitting through this training to learn how to do it better and to be able to keep this going. Yeah, so I think we're pretty much wrapped up here for today, unless anyone has any other questions. And if not, our next training is on Thursday, same time, just to go over our protocols for sound toxins specifically. I'm not sure if there's non-SoundToxins folks here. I mean, you're welcome to come, but it'll be mostly what our program does. And then we'll have our in-person training on the 18th. If you're able to come in person, please bring a compound microscope if you're able to. And we'll be doing just a lot of like hands-on in-person stuff. If you can't come in person, we'll hope to stream it virtually. And the second day on Wednesday, the 19th, will be our meeting. We'll have a bunch of presentations from a bunch of different folks. So we also hope you'll be able to join us for that. And it's the same Zoom link for everything in case you're joining virtually. So you can use the same link for all 4 of those or sessions. Any last questions? Okay. Thank you all so much and hope to see you again later this week.